Bismillahirrahmanirrahim. Assalamu alaikum, dear student. My name is Dr. Muhammad Adnan Asgar, and today the topic of our discussion is characterization of pharmaceutical materials by using thermal techniques. So far, we have studied some of the important aspects of the said topic in our previous lectures, and today we will discuss some major and important aspects of drug delivery system by using polymer microspheres. So, drug delivery system using polymer microspheres. Until now, we have focused on diffraction scanning calorimetric analysis of individual amorphous or sometimes crystalline drugs in pharmaceutical analysis by using thermal analysis. We will now apply what we have learned from those experiments to a much more complex sample consisting of amorphous and hydrated crystalline drug, which will be dispersed in some of the biodegradable polymer microsphere. It usually of 50 to 50 to 200 microns. For example, the TGA results, thermogravimetric result in this picture actually shows the sample losses just over 2% by weight at about 150 degree Celsius. Therefore, it is necessary to use hermetic pans for the diffraction scanning calorimetric experiments. The next step in determining the crystalline content of the drug in the microsphere is to run at different heating rates. The next step is actually for the determining the crystalline content of the drug in the microsphere is actually to run at different heating rates. That would help us to see if the sample undergoes polymorphic transformation at low heating rates. In the next picture, it is actually a comparison of total heat capacity signals from experiments run at 1, 10 and 50 degrees Celsius per minute. Results show that only 50 degrees Celsius per minute is a single possesses a single melting peak. This means that the sample must be run at 50 degrees Celsius per minute in order to measure the original crystalline form of drug instead of other polymers that form at slower heating rates. So, 50 degrees Celsius per minute is a quite reasonable heating rate for the successful identification of polymorphs in this crystalline compound. At about 50 degrees Celsius, here we can see that in this picture the first heat shows a total heat of fusion of 12.58 joule per gram, with 97% of that coming from the peak near 115 degrees Celsius. Since the pure drug has a heat of fusion of about 98 joule per gram, this means that the microspheres contain about 13% crystalline drug. To confirm reproducibility of the measurement at 50 degrees Celsius per minute heating rate, the sample may be run in the triplicate with the excellent results that is actually showing. In addition to the crystalline drug, the microspheres contain an even higher concentration of amorphous drug. However, all the data that is shown in this picture only a single glass transition at about 35 degrees Celsius. This means that the amorphous drug and the amorphous polymer of the microspheres are completely miscible, and it is not possible to measure the amount of amorphous drug in the sample by diffraction scanning calorimetry. That is another approach, such as thermogravimetric analysis, somehow needed. TGA or thermogravimetric analysis data of some of the compound will show that the polymer microspheres, it should not be drug, are usually essential fully decomposed at near 400 degrees Celsius, where the rate of weight loss is minimum. This is in contrast to the microsphere with the drug, which are still losing weight as a relatively high rate due to ongoing decomposition of the drug. So the next one is the drug-excipient interaction. Actual drug dosage forms are seldom just the pure drug or protein. Instead, they are usually composed of multiple ingredients that aid in the manufacture, storage, or delivery of the active ingredients. Because the dosage form must be stored over a period of time at some temperature and relative humidity, so there is a need to confirm that the efficiency of a drug formulation will not change with time. 2 days used by pharmaceutical companies to improve storage stability or to keep the sample below the glass transition of any gradient. It actually minimizes molecular mobility and therefore possible chemical interactions. It is also help us to use crystalline drugs that are more thermally stable than amorphous drugs. So in generally, the drugs should be kept below the glass transition point, because uh, above the glass transition point there is a possibility of molecular motions. The molecular strong molecular motions may excite the some part of the drug or the molecule, and it may help to interact with the other impurities or some moistures present in the atmosphere. So that would be quite helpful. The glass transition would be quite helpful for to understand the stability and the reactivity of the specific drug. It is usually the responsibility of the drug formulations group to determine whether the drug will interact with any of the other ingredients in the final formulation. It's very important to learn the formulation group to determine if the drug will interact with any of the other ingredients. This is often a very tedious task where multiple samples must be stored under different conditions for long periods of time and tested regularly with a variety of analytical techniques. Diffraction scanning calorimetry has proven to be an excellent tool for detecting drug excipient interaction. A fingerprint of a fresh formulation is made and then compared to the aged sample to look for differences in the transitions of any ingredient, especially the active compound. So it means that the diffraction scanning calorimetry technique would also help us to compare the freshness of the sample. It would easily we can get the desired data easily by just comparing the diffraction scanning calorimetric data of fresh sample and the aged sample, and this would help us to give a quality of a drug in a more specific way. For crystalline drugs that melt without decomposition, this is a relatively easy measurements because the peak area of the melt is a quantitative measure of the crystalline drug. For amorphous materials that are often miscible with other amorphous excipients, or for the crystalline drugs that decompose instead of melt, the measurement of drug excipient interaction by diffraction scanning calorimetry is somehow difficult. Just as with many of the previous examples, always start with the analysis of new materials with thermogravimetric analysis TGA. This will save a lot of time in the end and definitely help to provide the correct interpretation of many transitions. This figure actually shows thermogravimetric analysis results on a cold or allergy tablet, and it is written over there. This is the cold energy tablet that used as a crystalline drug as active ingredient. Analysis of the weight and derivative curves shows the sample contains more than 1% volatiles and subsequently starts to slowly decompose above 100 degrees Celsius. It is quite obvious in this picture, which is actually the thermogravimetric curve of cold, or we can say allergy tablet. This means that diffraction scanning calorimetric experiments need to be performed in hermetic pans in order to avoid a large endothermic evaporation peak that could hide other weakened in transitions. While the diffraction scanning calorimetric data for this material is shown in this picture. This is actually the first one is actually the more emphasis was on the thermogravimetric analysis. We know that the thermogravimetric analysis is actually the combination of the mass change with reference to the temperature enthalpy. But in thermogravimetric analysis, we usually observe the mass change with respect to the temperature. The DSC, diffraction scanning calorimetry data of this material is shown in this figure, which is actually is a comparison of three separate heating experiments at 10 degrees Celsius per minute on the same sample. The first heat, here we can see this is for the first, and this is for the second heat, and this is for the third heat after drying sample at 150 degrees Celsius by placing a pinhole in the pan. So first we would like to talk about the first heat in the sealed pan. The first heat in the sealed hermetic pan does not show any transitions until just below 100 degrees Celsius, where the sample is known to decompose after the TGA curves of this. Here we can see this is the TGA curves of this sample. The endothermic peak between 100 and 150 degrees Celsius could easily be misinterpreted as a melt if it were not for the TGA data. After the first heat to 175 degrees Celsius, the sample may be cooled and heat a second time in the sealed hermetic pans. Volatiles are not lost in this case. This run shows only a glass transition just above 0 degrees Celsius. This is actually for the second heat in sealed pan. Uh, the when the drug was if the drug was crystallized, it to begin with no glass transition but has converted to amorphous form as a result of the decomposition that occurred starting at 100 degrees Celsius on first heat. When evaluating storage stability of this formulation, the researcher should look for the formation of glass transition over time. One question that still needs to be answered is actually the actual temperature of the glass transition. This is quite important question to think and learn about the answer of the compound. As discussed earlier, the temperature of a glass transition decreases with increasing amount of moisture or solvents. The TGA results show that sample probably contains slightly more than 1% of water molecules. The thermogravimetric results shows that sample may contain slightly more than 1% of water molecules in the pharmaceutical sample. To determine the maximum glass transition of a dry sample, a pinhole should be placed in the lid of the hermetic pan at the end of second run, and the sample should be dried at 150 degrees Celsius in diffraction scanning calorimetric cell for 30 minutes. After drying, the sample should be heated a third time and the sample glass transition seen starting at 50 degree Celsius. Here we can see this is quite obvious in the third heat and ending near 100 and 100 degrees Celsius. Here it should start from the 50 near 50 degrees Celsius to 100 degrees Celsius. Therefore, the analytical chemist that is evaluating the storage stability of the formulation would look for the development of development of a glass transition between 0 degree Celsius to and 100 degree Celsius as the sample ages. The wide temperature range is due to the possible variation in moisture from batch to batch. This is what the traditional DSC and the modulated DSC can perform different operations, and it gives the accurate results. And we know that traditional diffraction scanning calorimetry provides a single signal which is the sum of all thermal events occurring within the temperature range of the experiment. This often makes it difficult to interpret data, or detect small transitions. While in the case of modulated diffraction scanning calorimetry, it has a significant advantage over the traditional diffraction scanning calorimetry in that it measures both the heat capacity component and the total and obtained kinetic component from the difference. Separation of complex transitions into the specific components greatly improves the interpretation of results. In general, modulated diffraction scanning calorimetry provides some of the advantages over the traditional diffraction scanning calorimetry. The first advantage of modulated diffraction scanning calorimetry over conventional or traditional diffraction calorimetry is the increased sensitivity. It is it have great sensitivity and have quite good resolution power. Moreover, the separation would easily separate the complex transitions. Moreover, accurate measurement of crystallinity in semi-crystalline materials and direct measurement of heat capacity while other while programming temperature or holding it isothermal. So increased sensitivity and the resolution is the quite important advantage of modulated diffraction scanning calorimetry over traditional diffraction scanning calorimetry, because it would help us to measure the small thermal events that occur during the temperature scan of that particular sample from its melting point to at glass transition temperature. It would even it would even calculate and observe the small thermal events that may be occur in the form of endothermic and exothermic. It would also help us to give the magnify the peaks of exothermic and endothermic in whole scan region. So the major advantage of modulated diffraction scanning calorimetry over conventional diffraction scanometry is actually the increased sensitivity, increased resolution, separation of complex transitions, more accurate measurements of crystallinity in semi-crystalline materials, and direct measurement of heat capacity either while programming temperature or holding it isothermal. So these are the some of the major advantage of modulated DSC over the traditional DSC. Uh, Here is the reference of this lecture. You can consult the book, which is the Thermal Analysis Fundamentals and Applications to the Material Characterization by Ramon. And the second one is actually the publication is a scientific paper named with characterization of pharmaceutical materials by thermal analysis by Thomas. This is all about this lecture. Thank you very much.